When will the exams be graded? I would love to have them graded by Friday, but I have another class also with 40 exams. I have 80 exams to grade, so probably not going to happen. I probably won't be able to hand them back until, yeah, although what I probably will do is grade them electronically, put my marks on one of these screens, and then I can email it to you hopefully next week. So, just 2 right now. Not this semester. For undergrads? It depends. Sometimes when I teach undergrads, I give Scantron exams. Those are wonderful. All right. So let's see. A couple comments. The exam, there was obviously not enough time to finish the whole thing. I haven't looked at it average yet, but I'll be surprised if anybody, completed it 100%. For next time, I'm talking to our tech folks about options that would enable you to have more time, or for me to give you more time, because those were all questions that I would like to have been able to get you to give me answers on. All of the questions that were on the exam were relevant. It's just that 50 minutes just goes by real fast. So one of my colleagues, he is able to do 70 minutes by saying you can come 10 minutes early and get started, and I'll let you stay 10 minutes late. We could do something like that. There's also for people who are following along at a distance, they take the exam with a proctor. So, like, find a boss or something who can sit in the room with you and sign off that I watched this person take the exam. They took X amount of time. So the timestamp is not an issue. It's by themselves and using the resources, only the resources that they've been permitted, which in our case is not an issue because you can use whatever you want. But generally, for people who are taking exams at a distance, we require a proctor of some sort. One thing that is possible in that regard is there are electronic proctors. So if you have a webcam on your laptop, you can pay $25 and log into this website, and somebody will be watching you while you take your exam. And you set up like a little, you have to set up like a mirror or sunglasses or a CD or something so that they can see a reflection and see that you don't have notes on the outside of your laptop screen. But that way you could take it at home, and I could give you an hour and a half or however much time we wanted to do. So, possibility. I don't think so. Yeah, it'd be a tough job, right? All right. Let's see. So next topic I was going to talk us through is next generation sequencing stuff. I think we'll do next gen sequencing for a few lectures. And from there, we'll either go to GWAS stuff or predictive modeling, statistical learning. So people have expressed interest in that, which might look like, let's predict, the species given sequencing data in a community of bacteria. Or let's predict whether you have cancer based on a microarray that we obtain from you, stuff like that. So let's talk about sequencing a little bit. We did already talk about sequencing early on in the semester, so we are familiar with the idea of taking genomic material like DNA and trying to figure out what the sequence of A's, C's, T's, and G's is that makes it up. That technology has been around for a long time. Sequencing has been around for a long time. But the technology that has been for the longest is expensive, time-consuming, and does not scale well to, like a human-sized application. But 10 years or so ago, I guess, they had new breakthroughs in the technology, which allows for much faster, much cheaper, much higher throughput sequence analysis of samples. And while this is fundamentally a sequencing technology, and you can use it to do a variety of things, not just figuring out what the ACs, Ts, and Gs are, but we can use it to do things like we were doing with microarrays, for example. Let's quantify the activity level of genes. We can use it to look throughout the genome for locations that transcription factors bind to regulate expression of other genes. We can use it to locate single locations along the genome that vary among individuals within the population. Lots of different things that we can do with just this sequencing stuff. So let's talk a little bit about the next-gen sequencing idea. I am going to refer you to this paper. I like this paper by Metzger. So this is on eCampus. It's a Nature review paper, Nature Genetics review. Okay, so I'm going to stop at a couple of places throughout the paper and just make some comments. So the second page has a section called Template Preparation. He's going to walk us through the process of doing this next-gen sequencing. The idea is we want to take genomic material, DNA, and we're going to break it into many, many small pieces. Basically, taking an entire DNA segment or a long fragment of DNA and trying to figure out the sequence does not work well in a high throughput application. But it can work well to take your DNA and break it into lots of little pieces, small little pieces, and then try to read or sequence the little pieces. So that is what happens. DNA is randomly broken into smaller sizes. This random breaking is important because we want, the randomness is important. So the goal is to randomly break the DNA into small pieces and then take those small pieces to sequence. Okay, sequencing and imaging. Let's see. Figure 2, I wanted to look at as an illustration of the next step. So step 1, we have DNA that we obtain, maybe from a tissue sample or a scoop of goo from a lake where there's a community of bacteria living or something. We want to take the DNA and break it into small pieces. We're turning our DNA into a collection of, quote, templates. Short fragments to be sequenced. Once we have those, we're ready to actually do sequencing. And let's look at figure 2 to understand the sequencing step. There's many different specific technologies out there that do what could be called next-gen sequencing. There's different ways to do sequencing fast, cheap, on a high scale, or on a high throughput scale. Last time I checked, the most widely used platform is Illumina. And let's look at the basic idea that drives Illumina. It's informative so that you know where these data come from. So we take tissue sample from something, say, and we extract DNA. We break the DNA into lots of little pieces, and we bind the little pieces onto a chip. So we talked about microarrays, those little chips that you can physically stick RNA to. It binds to complementary strands, the little carpet that we talked about on the array. Something similar is going to happen here. We're going to have a plate that has millions and millions of little carpet strands where DNA fragments can bind. When a fragment binds, to a place on the device, flow cell is what it's called, it can be amplified. So if there's a fragment that came from this particular location in the DNA, and we broke it into the little template pieces, and one of those pieces goes and binds to this device, we can amplify that piece so that we have a collection of representatives of the same thing. So here's one little read that came from our DNA. It's going to come stick to the flow cell device, this sequencing assay. And whenever things stick to the assay, we're going to kind of amplify or blow up, make multiple copies of strands that are bound, so that we have lots of representatives of the same sequence, ideally, in the same place. Here's a picture. So here's a device called a flow cell, and we're going to take our DNA fragments. So here are, can't change the color. Here's our little fragments of DNA, A, C's, T's, and G's. These are going to come hybridize or bind to the flow cell. And here's one of them that bound right there. What's going to happen is that we're going to get many copies made of that one in that little location. Here's another one that bound right here, and we're going to get lots of little copies of that one. And here's another one right here, lots of little copies of that one. So we're going to call, we have like these little clusters of representatives of the same fragment from the DNA. And we're going to have millions of them on this flow cell. So what we would like to do then, once we have bound our fragments to this device and amplified into these little clusters, we'd like to go to each of these clusters and figure out what is the sequence. That green little cluster, here's a bunch of representatives of the same sequence. Read depth will speak to how many reads, how many copies. If you have more amplification, you're going to have more read depth. Yeah, because there's going to be more copies of the same short sequence that were observed. The more representatives you have, the more opportunities you have to actually observe them. Right, so we want to go to each of these locations and figure out what is the sequence. Here's a picture that kind of illustrates the idea. Basically, we are going to add one base at a time. We have these little template strands sticking off the device, and each one of these little strands, ideally, is the same short DNA sequence. So what we could do, since DNA likes to be double-stranded, A's are friendly with T's, C's are friendly with G's. So if on one of our little strands that's bound to the assay, if the first letter in the sequence was, say, a T, then if we had a bunch of C's floating around, they should go and bind to that spot. So here, sticking to the device is one of our short fragments. The gray part actually is the DNA sequence fragment that we want. The red and the blue are special pieces that are put on the ends of our fragment to make it so that we can stick it to the device, and from the top so that we can start sticking individual bases to it and figure out the sequence. But the gray piece right here is what we want to sequence. So we're going to put into the mixture a bunch of representatives of the A's, C's, T's, and G's. And each of these representatives have been labeled with a fluorescent dye. We talked about that trick in the microarray case where you take RNA and you stick it to a chip, and because we have incorporated a fluorescent dye, we can shoot a laser at the chip, and then the light intensity tells us about how many copies of RNA are stuck in each cell. In this case, we're going to label the A's, C's, T's, and G's, and the technology allows for us to add a single base. We have these strands, these template strands that we want to sequence, and step one in an iterative process is to say, go put the first base in place. So on this strand, the first base that was added was a C. Here's another, a different strand. So this would be like a different cluster on this picture. If we went to the next cluster and mixed in these A's, C's, T's, and G's, and only allowed one of them to stick, this case, it was a G. In the third cluster, the first base was a T. So that ends this chemical reaction. We have all these A's, C's, T's, and G's, and using chemistry, we are able to say, only put one base in right now. What you can then do is take a snapshot. Shoot the thing with a laser to turn the fluorescent dye on, and then take a snapshot. So this panel right here is what we would see after doing that first iteration. The first base only across the entire assay. So all these dots are representing different clusters on this device. And for example, that green dot that has a circle around it right here, green means C. So the letter C has been labeled with a green dye, fluorescent dye. So when we shine a laser at the flow cell, wherever a C has been incorporated, will shine green. This particular spot we know corresponds to this particular fragment. The first base in this fragment is C. But then simultaneously, I have the first base in every other short fragment that's on my flow cell. Here's another one that has a C at the first spot. Here's one that has a G at the first spot. Blue is A at the first spot, and so on. So we have all this DNA, chop it into small pieces, stick all those little pieces onto a device like this. We mix in A's, C's, T's, and G's that have different labels on them. And I'm out of colors. That was the first iteration. I now know the first base for all the reads on this array, and there's millions of them. Then you go step 2. So now you manipulate the chemistry, put in another, or the A's, C's, T's, and G's are still there, but you manipulate the chemistry to say, add one more base. Take another snapshot. So for that first fragment, the first base was a C, because our first snapshot shone green in that location. The second base apparently is an A, because in the second round, when we imaged, we got a blue color at that location. The third is a T, the fourth is a C, and so on. So if you look down below those 6 snapshots, it says top was CAT, CGT, and bottom was straight Cs. So there were 2 spots consistently highlighted in these 6 images. The top one went green, blue, red, green, yellow, red. So that's CAT, CGT. We have just figured out a short fragment of our DNA. We've figured out its sequence. And again, on this flow cell, there's going to be millions of those little reads. So at the end of many iterations, we're going to have millions of little short reads. Okay. So we're not done, because all we have are these short little sequences. So say we're studying mice. We have tissue samples from mice under 2 conditions, and we've gone through this process that we just described. And now for each mouse, mouse 1 from the placebo group, I have a huge file full of short reads. If I wanted to compare the mice, like let's compare placebo to treatment in terms of maybe gene activity level or expression level, what I would need to do first is take those short little sequence reads and figure out where they came from in the mouse genome, because we don't have that yet. So here's the mouse genome. Here's all the A's, C's, T's, and G's for a mouse representative. And when we took our little tissue sample from mouse A, tissue sample, and ran that through the sequencing technology, the actual chemistry, the laboratory component, out from the lab. So here's my laboratory lab. We get millions of short little reads. So for this mouse I, we would have a short little read, have another short little read, millions of them. But there are, here's our mice that are placebo, here's our mice that are treatment, and we would like to compare what's going on genetically between these 2 conditions, control, treatment. In order to do that, what I need to be able to do is take all of these reads and bundle them, group them in terms of their locations in the genome. So instead of this little list of short reads, what I would like to do instead is say, I saw in mouse I saw 35 representatives of that region of the DNA. So I saw a short sequence that was right there. I saw another one that was right there. It overlapped. Here's another one that is right there. Here's another one that's right there, and so on. 35 a count of 35 for that particular location in the genome. For the next one, maybe it's 120 copies, and maybe for the third one, it's not very many, it's only 20. If we could do that, then call this region 1 or gene 1, region/gene 2, region/gene 3. Then we can have a data set like we're familiar with now, an M by N data set, because I can have a separate row for each of my regions. Here's a region, here's a region, here's a region. And then a separate column for each of my mice. For mouse 1, region 1, I'm going to put 35. And for mouse 1, region 2, I'm going to put 120, and so on. So now we can have an M by N matrix of numbers. Each row is a gene, roughly speaking, and the numbers represent, they might represent activity level if this was RNA copies that we were tabulating. Once we have that matrix, we can do things like we've already done. Let's do statistical tests to ask about differences between treatment and control. Let's do clustering, let's do PCA, stuff like that. One thing that will be interesting in the context of next-gen sequencing is that our data are counts. They are integer counts. They're not continuous intensities like they were for microarrays. How many reads did we see out of that big pile of reads that we deduced in the laboratory? How many of them would we say overlap or came from or aligned to or mapped to that region? They don't have to be the exact same sequence as long as they are substantially overlapping with a common sequence, a common location. So at this point, in the paper at least, we have a big old pile of reads. We had our tissue sample from a mouse, ran it through the instrument, got back out huge pile of reads. That might be in a FASTA file, by the way. We talked about a FASTA file earlier this semester. That's a simple, standardized way to pass along sequence information. So here's a huge FASTA file for mouse 1, and inside is a separate little entry for all the millions of reads that we saw. That's where we are right now. Next step is to either align our pile of reads back to a reference version of our genome, or something I'll talk about in a second. But this stuff, this mess here, is referring to alignment. I have a pile of reads from a mouse. It turns out that for mice, we know the genome. Somebody or somebodies have put in the work to figure out the entire genomic sequence for at least a representative mouse. And as we've said before, the genome does not change substantially across representatives of the same species. There's a lot that remains unchanged. So we could use a representative of a mouse. Here's a sequence of a mouse. Here's all of my reads from this mouse. Let's figure out where they came from in the original mouse genome. That's alignment that we've talked about before, sequence alignment. I want to take, say, this read right here, and I want to compare it to the entire genome, the reference genome for a mouse, and align it to that. That's what this is referring to, alignment. Once you have your NGS reads, that's what the pile of short sequences are referred to, what we need to do one of two things. The first is what we're talking about right now. I have there's an organism I'm studying, and I know the genome for that organism or for a very closely related organism. In that case, I can take the reads from a representative of that organism and align them back to the reference. That would allow me to get this, X. Because for the mouse, I can say gene 1 is located right around here. It has sequence such and such. Gene 2 is located over there. It's got this sequence and so on. So then we could look through these fragments, align them back to the genome, and if they overlap with gene 1, we can say that's +1 for gene 1. That's one copy that I saw for gene 1. Here's another read that aligned and overlapped with gene 1. Now it's 2. ... You're counting up how many representatives of a common region were observed. So alignment is commonly done. People on campus that I've talked with who are doing next-gen sequencing, most of the time are doing this alignment stuff. So they're studying cattle, for example. Let's breed cattle. Or let's study, what else have I seen? Microorganisms, so bacteria, certainly humans. There's a reference genome a lot of the time. And they do next-gen sequencing to get reads from samples. They align them back to a reference genome, and then we do statistics on this matrix of read counts. That's what you're going to do in your homework, your next homework. I'm working on that one. But you're going to download some data, which I have on this computer now. Let me show them to you. On eCampus, under data NGS, so there's a separate folder for next-gen sequencing data, and then this folder called Hammer. These data come from a paper with first author Hammer. Here's what they look like. There's two files. One of them is where the bulk of stuff is. This one is kind of like metadata. So let's open up the phenodata file, text file. So we are going to have 8 samples. We're going to have an M by 8 matrix eventually of read counts. And these text entries are telling us about the meta information for each of the columns. I guess I'll show you more. I'll give you more detail on this when I post the homework. But there's a control and there's a treatment. There's a time as well. So each of these mice, these are mice, and each of them is either treated in a usual way or they are, I think their brains are affected somehow manually to induce chronic pain. And then we sequence them. We do next-gen sequencing on them at 2 months and at 2 weeks. So what is the expression level differences under this treatment protocol? So under this type of disease, basically. This is something that they do to mimic a disease. And how does that change over time is kind of what we have. Look at the raw data, the actual numbers. These are not raw data, I should say. These have been processed substantially because somebody had to do this for us. This is what comes out of the laboratory, and somebody has to turn those into capital X. That's something that's kind of important to recognize about next-gen sequencing. It's different from the microarray stuff that we talked about in the first part of the semester. The microarrays are also can think of as like a chip or a plate, a device, but we pre-engineer, or the company pre-engineers, these arrays to have these wells, and each well corresponds to a known quantity. So this might be gene X with known sequence for the mouse, A, C, T, G, A, ... So before we even get our tissue sample from the mouse, we order this from Affymetrix, and we get it back in the mail, and it's got all these spots, and in the documentation it says row 1, column 4 on your array corresponds to gene X. Whenever you do your experiment and you get a light at that spot, that's your light for gene X. In next-gen sequencing, we don't have a pre-constructed device. There are no known homes on this flow cell. There's not a well for gene X. So there's a big additional step before we can actually get data that we're ready to do PCA, clustering, hypothesis testing, FDR, and all that on. There's a big step that has to be taken, which is to identify. Here's a little cluster of common sequences. This is a little read that we found, short sequence from the DNA. We amplified it, so we got lots of representatives of it. So here's something. Here's a thing, a read that I would like to get, but I don't know, is that gene X or gene Y or what? This alignment back to a reference genome, that's how we, quote, identify. Here's a read from my tissue sample. Is that gene X or gene Y or gene Z or what? We align back to a reference genome, and that's how we identify. Somebody asked just today on the discussion board also about mass spectrometry. Mass spectrometry is used frequently in proteomics. Proteomics, you're doing similar things. We have thousands of genes, and we want to quantify their activity levels. Proteomics, we might have hundreds of proteins, and we want to quantify their activity levels. Remember, with the central dogma of molecular biology, we could look at the DNA, we could look at RNA, we could look at proteins. Those are all valid places to look to understand what's going on. So I could try to quantify activity level or copy number, basically, of proteins, and we would do that possibly with mass spec. Mass spec has the same exact issue that we're talking about here. There is no device like this with predefined wells sitting there waiting for protein A. There are actually some chips like this for proteins, but they're not as widely used as, say, mass spec. In mass spec, you have a similar issue. You are going to see not sequences in that case. You're going to see masses and charges and stuff like that, but you see something, but then you have to match that to either some kind of database of known quantities. You have to identify what you actually saw. Just an FYI, you should always be aware, especially if you're going to have a statistician on your business card, you should always be skeptical and ask lots of questions about where did the data come from, because it's important. Here's my data matrix that somebody gives me, Excel spreadsheet, 50,000 rows, 20 columns. Somebody did a next-gen sequencing experiment. Row X says gene such and such, and we need to just be aware that there's uncertainty in whether it really is gene X because of this process. We had to figure out, based on what we saw from the experiment, which gene that corresponded to. Okay, so here are the data that have been processed. These are the Hammer count table data. Next-gen sequencing, people will talk in terms of read counts. We're getting reads. That's all we're doing. We're sequencing, and we're getting these short little sequences. When we align back to the reference genome, we're able to say, for example, in mouse 1, there were 227 of our short little reads that overlapped substantially, overlapped enough for us to assign it to a gene with this ensemble ID in the mouse. In mouse 2, it was 212 copies, or count, 229, and so on. So these are our numbers. Instead of continuous intensity measurements like from microarrays and other technologies, we have these counts. Okay. So that's where we're headed. We're going to do analysis with counts. We can do similar things that we've already done, like clustering, PCA, FDR. One thing that's going to be a little more complicated is how do we do statistical testing. We've done t-tests, we've done regression. Here we have numbers, but they're counts. And there are some important differences between doing inference on continuous numbers and doing inference on counts. So there's a whole bag of specialized methods, statistical methods, that people use for next-gen sequencing. I'll show you some R packages that have been developed. Basically, we want to do t-tests and regression, but in order to be careful, because of the special nature of these data being counts, those aren't perfectly applicable. They're not well suited to count data. But we can do something that is suited to count data. So we're going to do basically t-test-like things, regression-like things, but it's going to be customized for count data. And the good news is there's R packages that will do that for you. I will show them to you. All right, let me try to get through this paper. There's just a couple more things I wanted to say. Usually, at least in my experience, there is a reference genome like we were talking about. We're studying the mouse, and we know the mouse genome. We're studying the human, we know the human genome. Horse, cow, lots of things you know the horse, or you have a reference genome. Sometimes you don't, though. If you're studying a sequence or an organism that has not been fully sequenced yet, and there's lots of those out there, somebody has to figure out the original sequence at some point. And if we have this pile of reads, but we don't have a reference genome, we can still try to recover the reference genome. So if I had all these reads, is there a way to assemble them into an entire contiguous collection of sequences? Hopefully it's clear that that's a harder problem than aligning to a known genome. Aligning to a known genome, that's like that pairwise alignment stuff that we talked about, or multiple sequence alignment stuff. De novo assembly means assembling a genome from, what does de novo mean? Who's our Latin people? Okay, so it means without a reference genome. I'm wondering, like, what is that Latin or what? I forget. I think from nowhere, out of nothing. Right. Yeah, that's what I thought. But I wanted somebody else to feel stupid if they got it wrong, not me. Okay, so if you don't have a reference genome, you can try to assemble one from your pile of reads. Much harder, much more computationally intensive, but a valid task if you don't have a reference genome. Okay. Let me go to one last section, applications. I've mentioned this before. Next-gen sequencing is just sequencing, which has been around for a long time, so what's the big deal? The big deal is it's super cheap, relatively speaking, super high throughput, which means you can get lots of data out, and it's fast. So it's like a really nice, sophisticated tool, and we would like to figure out every which way we can use that tool and exploit it. There are many ways that we can exploit it. Rna Seek, you'll hear that bandied around, people who are doing research. Rna Seek is, think of that as next-gen sequencing's version of microarrays. We're going to, the reads, the counts that we're going to get, these counts, are counts of RNA representatives of gene X. That's what we basically were doing with microarrays, except we didn't get counts, we got continuous intensities. Brighter intensities at spots corresponded to more copies, more RNA copies, which means greater activity level of this gene. Here, we're actually trying to count representatives mRNA copies floating around. If there's lots of copies of the gene, there's going to be lots of proteins of that gene, because that gene's being highly expressed. So certainly one thing we can do with next-gen sequencing data is transcription analysis, differential expression analysis, like we've done with microarrays. That's referred to as Rna Seek. There's several different X-Seek flavors that you'll hear about: Rna Seek, Chip Seek, Methyl Seek, DNA Seek, MySeek, all these different things. Rna Seek is like microarrays. Another application is called ChipSeek, where we are using, I talked about this before actually, where there are these transcription factors that are proteins that can bind to specific locations in the DNA and regulate nearby genes. How active do we want some genes in this area to be? Send in a transcription factor and it will turn up or turn down. So if you have a transcription factor that you're studying, you can design an experiment where you can sequence all the regions of the DNA where it binds. So here's my transcription factor. I'm going to mix in lots of copies of it with the genomic material from this mouse. Let it mingle so that the transcription factor goes and sticks to every place that it wants to. And then it is possible to pull out only the locations where the transcription factor is bound and throw everything else away. And then it's possible to kick off the transcription factor and then sequence all those little pieces. So at the end of one of these applications, instead of a huge pile of reads, we have a much smaller pile of reads. And those are specifically reads where the transcription factor bound. So if we map those or align those back to the reference genome, we can figure out where in the genome a transcription factor binds, which can tell us possibly about what genes it is involved with. If transcription factor X is known to bind right here, here's 10 genes that are nearby there. We might want to look at those genes because that transcription factor might be controlling those genes. Methyl-seq, DNA-seq, these are related to ChipSeek. They're using next-gen sequencing to look at other ways that you can have genetic regulation. I mentioned methylation as a way to turn down expression. Often DNA's similar thing. One last comment and then we're done. Metagenomics could look like you have that lake with a film of bacterial goo on the top, and it's probably a mixture of several different bacterial species. Let's take a sample of the goo, sequence it, and now we want to ask what species are there and how much of them relatively are there. It's these 4 bacteria, there's 20% of this one, 60% of that one, and so on. You can use sequencing data to do that kind of exercise as well. And that's handy, like, let's see, well, with this entomology data, takes scrapings from cadavers and wants to figure out the community structure of the bacteria. So you might use next-gen sequencing to do that. Okay, we are out of time, but that's basically my introduction to next-gen sequencing data. What we're going to do is spend a handful of lectures looking at some real data, and I will show you some of the R packages, bioconductor packages that are out there to deal with them. So there's 2 or 3 packages for doing RNA seek, there's one for ChipSeek, and so on. Yes? So why use read count, not and so you could look at presence absence of single locations where there is variation, but that's talking about variation. That's not talking necessarily about activity level. So if you wanted to quantify, well, if you were only looking for SNPs, yeah, I guess you could probably do essentially the same thing if you could quantify SNPs across the genome, and if it was so if you have SNP data, you have yes or no, presence or absence for each individual. So how does that tell me about overexpression, underexpression kind of thing? You're not talking about RNA seek though, again. Sorry. I guess I'm still seeing you would be limited in what you can do. If you have these reads, then you can quantify activity levels, for example. You could do a de novo assembly if you wanted to reassemble things. If you only had then SNP data would be perfect. Yeah. I will comment on SNP Seek, basically, is what people would call it, I guess. SNP Seek, where you're trying to, I'll have to read up on it because I'm not as familiar with it, but you're applying this next-gen sequencing and you're specifically looking at only locations where there's variation in the population. Yeah. Okay. Okay. I'll look for SNP calling techniques in the next-gen sequencing context and try to bring something into the classroom. All right. I think it's good. Okay. Thank you.